If you've ever lived in a wooded area, you're probably familiar with the nightly chorusing sound of a forest full of frogs. Frog calls are used specifically for attracting mates, so vocalizing plays a critical role in reproductive success and the health of a species. We wanted to know how man-made noise affects the calling of frogs here in Anastasia State Park in the Guantanamo-Matanzas National Estuarine Research Reserve.
Frogs vocalize the most during their breeding season. In Northeast Florida, most amphibians breed during the summer months. Our study was conducted from September to October, so we focused on species known to call during these months.
These species include the southern cricket frog, the little grass frog, the pine woods tree frog, the American bullfrog, the southern leopard frog, and the Cuban tree frog. The two species we encountered the most in our data were southern leopard frogs and Cuban tree frogs.
Southern leopard frogs are native to Florida. These frogs prefer habitats with shallow bodies of water like swamps and forests.
Listen to this example of a southern leopard frog call. Cuban tree frogs are invasive to Florida. They are native to Cuba, the Cayman Islands, and the Bahamas, but have been colonizing Florida ecosystems for decades. Cuban tree frogs prefer the high humidity of maritime forest habitats, but are often found on and around man-made structures. Listen to this example of a Cuban tree frog call.
We assessed the rate at which frogs call at two study locations. The first location was Anastasia State Park. Here we had 6 sites: the quarry, concessions, an unopened trail, a bridge on the Ancient Dunes Trail, and the fire line of the park. Because of Anastasia State Park's central location within St. Augustine, these sites were exposed to high levels of anthropogenic noise. Our second study location was the Guantanamo-Matanzas National Estuarine Research Reserve. Here we had one site in the woods south of the Guana Lake Dam. The reserve is more isolated from development, so this site experienced minimal anthropogenic noise.
Our equipment included 2 Wildlife Acoustic Song Meter Micros and a Song Meter Mini, loaned to us by the GTM Research Reserve Rangers. We attached these acoustic monitoring units to a tree at each site for a period of 5 to 7 nights. The devices recorded from 7 p.m. to 6 a.m., as most of our frog species are active at night.
In order to set the song meter up, the top cover was first popped off. Then the silica gel packet was removed, and 3 AA batteries were added to the appropriate slots. Then, a 32GB SD card was clicked into place. With the system set up, the song meter is turned on and we wait for the recording and SD status light to turn solid green. Status button is then held down for about 3 seconds or until the Bluetooth light starts blinking.
Going over to the Song Meter app, we find the correct song meter and pair the app to the device. Next, we configure the recording. We choose to leave all the default settings except for the recording time, which was adjusted to the desired setting.
Between our sites at Anastasia State Park and the GTM Research Reserve, we recorded and analyzed 450 hours of data.
When the recordings were collected, we exported the data and analyzed it in Wildlife Acoustics' accompanying processing software, Kaleidoscope. This process involved visually detecting frog calls in the software-generated spectrograms. We identified anthropogenic noise events in the spectrograms and calculated call rates for 10-second windows before, during, and after the event. At the GTM, where noise events were few, we calculated the number of calls produced over a 10-second window. Comparative statistical analysis were run in R to identify differences in the mean call rates of each population.
Frogs are already vulnerable to human impacts like habitat loss and water pollution, so anthropogenic noise has the potential to push frog populations past the point of recovery.
The findings of our study have management implications for healthy acoustic habitats in modern nature reserves.
Future studies could even expand upon our analyses and assess additional acoustic components of frog vocalizations, as well as spatial modeling of acoustic habitats at Anastasia State Park and the GTM Research Reserve.